Welcome, everyone. My name is Hans-Christian Mahler, and I'm going to present to you today about primary packaging considerations from a biologics product perspective. Before I dive into the topic, just here enclosed my regular disclaimer. Biologics are an increasingly important class of molecules. And if you look at biologics in development by product category, by different types of molecules, molecular classes, I think it's easy to see that there's a number of different products, both in the commercial stage or entering the commercial stage, or are in clinical phase 1, 2, or 3 clinical studies. The biologics products account today for around about a third of annual approvals, and more than 900 medicines are in development, targeting a variety of different diseases. Now, biologics are and have to be administered parenterally. That means by injection, infusion, or implant into the human or animal body to really achieve a systemic bioavailability. So to make sure that the drug really meets its targets. However, biologics are also increasingly complex and sensitive, and this provides specific challenges, including cost of goods, or the COGS, but also stability. Looking at the biologics R&D, so research and development, and the pathway from a new molecule from the discovery stage to submission and beyond, it's very clear that this is a multi-year process, starting with the selection of a molecule, or the so-called clinical lead selection, with preclinical testing, so toxicological studies, the first in human or entry into human phase 1 studies, moving into pivotal, so submission relevant studies, and finally submission and launch. Finally, even after commercialization, these products can undergo so-called line extensions, so improvements in the marketplace. Biologics do have lesser attrition than traditional chemically synthesized small molecules. However, even of biologics, only around about 15% of phase 1 molecules are expected to reach commercialization, which in other words means that 85% of the investments are not recoverable, which also means that, especially in early stage development, the gating of investments is an important consideration. So it's going to be very important on where to put money, especially into early stage development. The commercial product image of a final drug product, so this final sterile product that is going to be entering the market, is very, very different than the early stage product image that goes into clinical testing. This also relates that the early stage formulation, so the composition, will be very, very different, but also the choice of primary packaging is different for early stage, typically saying. So as an example, when you have a sterile product and the biologic, in many cases, a vial is being used for something like the preclinical stage, as well as clinical stage 1 and 2, whereas maybe a syringe or auto-injector for some indications and diseases are the commercial image that would then be introduced for or during a phase 3 and launch. There's a variety of components of the final drug product, and all of them constitute on the success of the final drug product. So the formulation or composition is one of the parameters that is very important. This includes the choice and selection of qualities and quantities of excipients, but also the choice of dosage form if we are speaking about a freeze-dried product, an aqueous product, etc. The control strategy, this links to the choice of analytical methods, the definition of specifications, both for drug substance as well as drug product, but also generally speaking for in-process control testing, for excipient testing, etc. So the choice of methods as well as the limits that we apply to them from a regulatory perspective. The choice of a primary packaging is of utmost importance, which may or may not be connected to the choice of a device. And I'll be speaking a little bit more about that. And last but not least, it's about the manufacturing process, the appropriate design, but also the flawless execution of a manufacturing process that really determines the success of a product. You can have a most wonderful formulation in a great primary packaging, but if you cannot manufacture, you still don't have a product. Coming to the choice of dosage forms. Now, when you're developing a sterile product, you can typically choose during development if you want to go down the road of targeting a liquid formulation, or you could target the lyophilizate or freeze-dried product, or especially with increasing awareness of the COVID vaccines, also frozen liquid products are increasingly used by some companies. Now, it's very obvious that the stability of the product in liquid state will be less compared to the dried state. However, the speed for development, but also on how supply chain will be manageable, the scalability and manufacturability of these products, as well as user handling, are all advantages of a liquid formulation. A liquid formulation is also then easily compatible with devices. So imagine a liquid product in a cartridge or prefilled syringe that would be used in a pen, auto-injector, or anything else. The freeze-dried product has some advantages with regards to stability. You may have advantages to speed. Supply chain definitely has some advantages because your freeze-dried product, typically speaking, is more stable than the liquid product. Having said that, lyophilizates typically have some challenge in scalability because there is an additional process unit operation, which is freeze drying. So this is less preferable. Lyophilizates require user handling because the user, which could be a healthcare professional or patient, needs to dissolve or reconstitute the cake and powder back into a liquid for administration. And these handling steps are pretty prone to quite some challenges. So in a nutshell, it's great from a stability perspective to choose a freeze-dried product. However, you need to reconstitute, so it's not the most user-friendly. You have limitations in batch sizes. You have additional technical risk. You have additional cost. And basically, you also need to be aware that it's less easily compatible with devices. So these kind of products would, for example, require dual chamber syringes or any other solutions like that, which by itself brings quite a ton of other challenges along. With regards to frozen liquids, obviously in the frozen state, products may be more stable. Having said that, it depends on the formulation, the final freezing step, the thawing step, the freezing temperatures, etc. And whilst it is generally believed that frozen liquids are providing some advantages with regards to development speed, I'm not necessarily convinced here. Supply chain definitely comes with a ton of challenges because you need to store and ship these materials in frozen state at preferably defined temperatures. Scalability is warranted from a manufacturing perspective because you're filling, still liquids. Having said that, you still need the whole freezer and storage capacity to manage that product. From a user perspective, you need to thaw these dosage forms or products in frozen that are delivered in frozen state, this also comes with, freezing comes with a couple of risks. For example, for a lack of homogenization or dehomogenization, which basically means for the user that he or she needs to also ensure homogenization of the products after thaw. Additionally, depending on the storage conditions and storage temperatures, and depending on the primary packaging choices, for example, the stopper TG prime, you may also experience potential container closure integrity issues or concerns that go alongside. making these products a little bit more challenging from a regulatory perspective. These products are also typically not finally quality controlled by the user after thaw, except for maybe making visual testing for particulates, as much this is doable from a user perspective. Containers can break during shipment. You need to have specific considerations from formulation, and all of that basically are things to be considered when choosing the dosage form. Now, the selection of the required container closure system, or CCS, pretty much depends on the final target product profile and the so-called quality target product profile, which depends again on the formulation, on the dosage form, liquid, lyo, frozen, depends on the intended storage conditions, depends on the stage of development, whether we are talking about an early stage or a late stage / commercial configuration. It pretty much depends on prior knowledge and experience, and it depends on the intended administration. So whether you are targeting, for example, for patients to self-administer, or if it's going to be healthcare professional managed, if you want to combine it with an auto-injector, subQ pump, or whatever. And then you really need to choose the products, the container closure system, but also the supplier. You need to be selecting the material, the exact materials, and compositions thereof, but also need to ensure that if you're selecting individual components, like for example a vial, a stopper, and a cap, that all these components really fit appropriately together, and that these combinations are also adequate to be manufactured as a combination. As an example for early stage, in an early stage development, the selection pretty much can be, for example, between a vial in different sizes, a syringe, cartridge, or other. And another key consideration for early stage specifically is that you need flexibility of dosing, because your dosing regimen and your finally to be commercialized dose is not appropriately defined. So you're testing different doses. The doses can be tested by different concentration strength or by different volume. Typically, it's facilitated by choosing different volumes. So you definitely need a product image where you can achieve different dose strength and dose levels really by dosing different volumes. In addition, you want to have a facilitated supply chain. So whilst you can manage with 10 different concentrations in a clinical study, it would also require you to manufacture 10 different batches of drug product and to manage them throughout all your clinical studies. So a facilitated supply chain would definitely be achievable the less different concentration versions you have, and the more you really focus on dosing by volume. Considerations of the TPP could also be an early stage consideration. So how much you want to early on introduce your intended commercial image. And obviously, you need to plan for your upfront and early on for any switches of the container closure system, but also formulations and or any other changes of your product. So for example, if you're going from a vial in intravenous administration in a phase 1, and you want to target a syringe for subcutaneous administration, you definitely need to plan appropriate switch activities, which include technical, but likely also in the example that I mentioned also includes preclinical, but also clinical considerations. Especially early, it may benefit for using standard primary packaging, so knowledge-based and platform-based, versus maybe more extensive screening of container closure systems, because this could become into a chicken and egg problem where you are defining good container closure systems, maybe based on your formulations, but after all, you should probably combine the selection of all these components in one go, rather than spending a lot of time and money upfront and early in development. A key choice to make is really the primary packaging material composition. And one of the most commonly used and traditionally used materials is definitely glass. It's safe, it's proven, Having said that, glass has a certain risk of breakage, so it could break during manufacturing on sterile filling lines. It could break during shipment, possibly during use, like in a, for example, clinical hospital setting. And another downside of glass definitely is its dimensional variances. So when you need to really have very tight dimensional controls, for example, in combination with specific devices, this is where definitely glass will have its downsides. On the other side, plastic material or polymer containers could be used. There's a variety of different polymers that are being used from a variety of different suppliers. In this case, you need to understand your extractable, leachable profiles. Then obviously these containers may and will likely show oxygen, so gas, but as well as water permeability. And finally, sterilization may be a little bit challenging because it may lead to discoloration of the containers and provide other challenges. In a nutshell, this is what usually makes plastic containers less preferable and higher risk for biologics, given that many biologics are oxygen sensitive, for example, and hence this may provide a little bit of a different risk profile. Primary packaging can also come in other variances. One example is basically that primary packaging can be provided as so-called bulk. Bulk primary packaging hence need to be washed and sterilized within the sterile fill and finish facilities, which may lead to variability of different processing conditions in the various facilities. And that could be quite relevant when it comes to sterilization, residual waters, the qualification of your washing process, the choice of sterilization conditions, and all these things may also matter when it comes to, for example, damaging the glass, taking glass surfaces, etc. Another choice for containers out there provided basically ready to use or ready to sterilize. So pre-cleaned, ready to use containers would in that case already be sterilized. Key considerations here are what is the desired cleanliness level? So many of the RTU containers, as one example, are definitely qualified in the test for particulates. Having said that, the particulates may be derived from different acceptance criteria than your final product. So this is a typical source of challenge. The method of sterilization and its residuals may have a potential product quality impact. So when we're talking about ethylene oxide as one example, for example, this may actually interact with biologics, leading to a variety of degradants. For pre-sterilized containers, you also need to consider the sterility assurance until point of fill. So until you really fill the containers in the fill and finish facility from the place of final sterilization, which means that sterility needs to be also guaranteed during the whole course of the supply chain, shipment and storage. And finally, there's some different considerations depending on the product image. So, for example, if you are using RTU containers in connection with lyophilization and capping, you may experience some specific challenges when you are processing in a nest versus maybe denesting and renesting. Further considerations for primary packaging material itself are really its coatings. So the benefits of such coatings may be to include specific challenges with glass that have been discussed in the past with glass delamination, particulates, glass leachables, and managing the risks of polymer containers. So basically improving, so having less oxygen and water permeability. Having said that, it's going to be important to note about the process and reproducibility of the production of such containers and also second sourcing opportunities. If you have coatings, you always need to worry about the stability, but also the safety of these coatings. So in essence, you need to adequately characterize your primary packaging containers on whether they're sufficiently stable in connection with your final drug product from a coating perspective. And the coating itself obviously needs to also be assessed in case that would leach or interact with the product formulation. Now, how do you qualify primary packaging for its use with a given biologics drug product? And that can be quite a lengthy process, starting with typical GMP activities, looking at supplier qualification, qualification and reproducibility of the primary packaging process, looking at long-term stability with the selected drug product formulation under real-time accelerated conditions, but also extended characterization testing. You need to look at washing and sterilization to the desired cleanliness level and derive adequate specifications when it comes to things like subvisible particulates, visible particulates, endotoxins. You need to ensure the manufacturability of the primary packaging containers on the sterile facility and lines. You need then to also consider on how the primary packaging materials would be captured by your existing media fills or aseptic process simulations, otherwise would further stimulate that. And last but not least, you need to qualify the container closure system as such, independently of the fill finish process. So looking at things like container closure integrity, capping quality, defects, etc., to make sure that you really have a sterile and tight container and product at the end of the day. When it comes to CCI, container closure integrity, assessing the fit of the individual components, also at the spec edges, is definitely an approach to and consideration to make. So you need to make sure that the selection per se will be ensuring a sterile product. You need to understand the impact of drug product processing. So things like capping, or in case of a syringe, plunger rod insertion, device assembly, may actually interact with your primary packaging container or final product and adversely impact, for example, container closure integrity. Environmental conditions like shipping and storage may impact container closure integrity. And finally, worth saying that the choice of container closure integrity testing methods and their validation is really key. So, for example, when we perform these CCI testings, we usually use the most sensitive PCCI methods like helium leakage, which is a really deterministic method using appropriate positive and negative controls with artificial leaks, ensuring high sensitivity for development and qualification. In a nutshell, different considerations may apply for final quality control. Now, on this slide, I was trying to provide a couple of examples where primary packaging related product challenges can occur. Things like there's precipitation, so visible particulates being observed in stability, which may be quite common, especially for sulfate-containing formulations. And potential root cause could be that the glass leachables like barium or aluminum react with formulation components like sulfate, but also possibly phosphate. And as a potential mitigation, you may choose the more appropriate formulation, or if your formulation is locked, to select, let's say, low leaching containers, like low barium containers for your product. In case of functional containers like syringes, you could see increase stalling or increase in injection forces, which is not too uncommon for low silicon syringes. And a root cause could be insufficient amount of silicon in the functional container or inadequate distribution thereof, which could also be caused by silicon leaching from the syringe into the solution as a function of time. And the potential mitigation is really to use sufficient amounts of silicon in the container. And then generally speaking, when it comes to particulates observed on stability, this could relate to silicon droplets. These could relate to proteinaceous particles and or glass elimination, where silicon droplets, for silicon droplets, the appropriate characterization is going to be important. If you're finding proteinaceous particulates, you probably need to look into a more appropriate formulation. And if you're seeing a glass illumination, you should really ask yourself if you have the most appropriate glass material, if processing like washing and sterilization is an issue, or if you have a specifically aggressive formulation, and or maybe you have all three of them. Just coming a little bit more into critical material attributes of prefilled syringes. So when you're having a prefilled syringe, you have a variety of different components here with rigid needle shield, the barrel, the rubber stopper, etc. And there's a variety of publications out there that discuss tungsten residues, for example, silicon oil or other material components that may relate to specific issues for a final product, ranging from protein oxidation, aggregation, particulate formation, allergies, or other incompatibilities. And I just wanted to provide this slide as a reference. There's a variety of mitigation studies and activities that I would be suggesting to look at to actually de-risk any of these issues that are maybe specific to a given choice of syringe. Last but not least, stability is not everything. So this is a slide discussing that injection time and hence usability is pretty much a function, obviously, of the product formulation, product choice with regards to viscosity, but also a use temperature. Having said that, also device capabilities, but also especially needle choice and needle inner diameter are going to be key. As well as plunger glass friction. So again, speaking about the siliconization here. So all these components would contribute to injection time. So it's going to be very important to consider also protein concentration ranges, temperature, use conditions, variations in needle inner diameter, etc., when it comes to failure mode assessments and quality by design. The future will bring a variety of different challenges to us in pharmaceutical sciences. So therapeutic modalities become more and more diverse. So we're looking at more complex proteins, gene therapy, oligonucleotides, cell therapy, exosomes, just to list a few. With that, different solutions for primary packaging will be required. There's going to be more focus on patients and healthcare professionals regarding delivery and usability. So drug delivery, drug device combination products like auto-injectors will be more common. We will see increasingly a focus on sustainability, very thankfully. And last but not least, technology and integration are going to play a more prominent role with telemedicines, tracking compliance, connectivity with apps and cell phones, and traceability of products alike. With that, I would like to summarize that biologics have to be parenterally administered, so hence they are a sterile product. A strong system thinking is required to integrate the interdependent systems and functions of packaging, formulation, the active pharmaceutical ingredient, the process, control strategy, just to mention a few. The future will bring more complex and diverse therapeutic modalities and dosage forms. And let's all focus on what matters most, which is the patients, to ensure efficacy, safety, and quality in treatments. With that, thank you for your attention. And if you want to learn more about 1023, you're invited to have a look at our websites. Thank you.